I'm very pleased to moderate this webinar. I'm Dr. Archana Bedi. I'm a physician. And we have a very eminent speaker today and a great orator. And I'm very glad to introduce Dr. Nitin S. Baliga, who is the professor at the Institute of Systems Biology, also known as the ISB in Seattle, Washington, US. He's a founding member and currently serves as a director and senior vice president. He is engaged in solving complex problems in global health, which is health across borders, personalized medicine, and the environment. His team develops predictive models of cellular and molecular networks of pathogens, cancer cells, and microbes in our environment. This enables biotechnologies to overcome drug resistance, find new drugs, and predict consequences of climate change. Dr. Baliga is a bachelor in microbiology from Ruya College, Mumbai University, a master of science in biotechnology from Goa University, a PhD in microbiology from UMass, Merced, Massachusetts, and further trained in systems biology. He has published more than 100 peer-reviewed articles in top international journals, including Cell, Science, and Nature, grossing more than 20,000 citations. He has an editorial role on several international journals. He has been the invitee to the Google Skyfu Camp and has been widely profiled in magazines and journals, including The Scientist, Genome Web, and many others. Dr. Baliga's research has been supported by NASA, the US Department of Energy, the DoD, the US Department of Defense, the DoD, US National Institute of Health, the NIH, US National Sanitation Foundation, the NSF, and Bill and Melinda Gates Foundation. Dr. Baliga also has made significant contribution to science, education, and outreach. Through the Systems Education Enterprises, the SEE program, he founded over 15 years ago, for which he has won the Alvin J. Thompson Award. The SEE program has impacted more than 2.5 million students and teachers across more than 100 countries. He is talking to us about personalized precision cancer therapy today. We all know that the process of drug development is expensive and time-taking. What we don't know is that it takes around 15 to 20 years and around $2.5 billion to find a drug for one given cancer. And still, this drug can be ineffective on majority of patients. This is because cancer cells are diverse, cancers are diverse. Even within a particular cancer type, the cancer cells can be genetically and phenotypically heterogeneous. Therefore, patients with a similar type of cancer may not respond or get treated with the same kind of cancer drug. Dr. Baliga will discuss strategies to formulate personalized therapy cocktails that precisely target dysfunctional gene networks within an individual tumor of a patient. I'm very glad to hand it over to Dr. Baliga. Dr. Baliga, please take it away.
Thank you, Archana. It's a pleasure to be here, and I appreciate everyone coming in. I think it's 7 or so in the evening there, so it's a real pleasure to be back in Pune, at least virtually. So I will speak about a topic that is on everyone's mind, about therapies not working as expected on patients, but with cancer as a focus. And you received some overview of this already. Let me just sum that in a couple of slides. So what I will do is I will, in 2 slides, tell you what the problem is and how we can develop a solution. Then I will go into some detail about how we can execute on that solution. And I'll then give 2 examples, one on brain cancer, glioblastoma, and the second one will be on multiple myeloma. So let's jump right in. So the process of drug discovery is a long and expensive process, and it's a very lossy process. It typically starts with the high throughput screening of millions of compounds. And you do the screening with cell lines for a given cancer. And the expectation is that if the assay you developed can detect a molecule that can impact the cancer phenotype of a cell line, then you can carry that on to the next phase and do deeper investigations. Now, the assumption there is fundamentally flawed in that the types of cell lines that are used are few, and they don't really capture the complexity and the heterogeneity of disease across patients. And even then, through the process, a lot of drugs fall through because they don't work on those tissues that are picked. And when the molecules that do work are discovered, they then go on through this very expensive and time-consuming process of phase clinical trials. And by the time you find a drug that is approved, you typically would have spent about 15 to 20 years and $2.5 billion on finding that drug. And the success rate through this whole process is less than 1.6%. So more than 98% failure rate. And the drug that is discovered, when that is used in the clinic, you find that in majority of patients, it does not work. And the reason being what I described earlier is through the whole process, even including the tissue that is used to find the molecule and the patients that are recruited to the clinical trials, they are not all consistent with the biology or the mechanisms underlying the different types of disease. The assumption is there's an average patient that represents the disease across a population, which is what is fundamentally flawed. But there is evidence that if you do know something about the underlying disease biology, especially as it relates to mutations in the tumor cells, then oftentimes you can find therapies that are effective against some patients. So you can then say that patients who have a particular kind of mutation, say the HER2 mutation in breast cancer, should receive a drug like Herceptin, and the response rate really shoots up. And the clinical trial success rate also increases by about 6.6-fold. So there is evidence that if we do understand the underlying biology of the disease, we can make progress in finding therapies that work on specific subsets of patients. But there's this assumption then, which is also flawed, that there's a linear map that links the mutations in a tumor cell to the responsiveness to drug. And those who have worked with these data sets know that tumors typically have hundreds to thousands of mutations. And in a given patient population, you typically have less than 10% of the mutations that are truly actionable, meaning that there's something that can be linked to the biology or the clinical response of the tumor through the mutation presence or absence. Now, the mutations don't act in a linear fashion. And just as we were wrong in saying that there's a one gene, one disease paradigm, this is also flawed. In reality, there's mutations acting in complex combinations with non-additive consequences. And as a result, a patient may have a mutation you think should predict response to therapy, and the patient does not respond. So what we need is a better understanding of genotype to phenotype and the networks that build that relationship between the two important information types. So this is what we've done, and this is a way that we can address this problem. There's this vast amount of information that exists already in the public domain regarding the biology of cancers, regarding how genes function, how they interact, and so on. And we've built a lot of these knowledge bases ourselves in my lab, including maps for transcription factor DNA interactions in the human genome, in the mouse genome, also microRNA target associations, and so on. Now, this knowledge base can be used as prior information or a guide to then mine cohort multiomics data sets from cohorts. When I say multiomic, that refers to transcriptomic, genomic, and all the different omics measurements that can be made in a patient's tumor, including clinical outcomes, survival, response to therapy, etc. The goal is then to navigate through these disparate data sources, and then through a combination of different algorithms, which I will jump into in a little bit, you then decipher the subset of mutations that have evidence of causally perturbing the flow of information through gene networks. And when I say networks, I mean regulators like transcription factors and microRNAs, controlling genes in modules that are associated with specific functions, including disease phenotypes, and have dynamics that are predictive of the outcome of response to therapy and survival and things like that. Now, a model like this has got causal and mechanistic underpinnings, meaning we can say A is there, therefore B happens. So that's causation. And we can in fact say A is there and therefore B happens through C, because C binds to B. That's mechanism. So we have causation and mechanism built into this network. What that allows us to do is then validate these models, meaning we can then precisely target genes within this network and say, if I were to perturb this particular gene, I would expect to see this particular outcome on disease phenotype. So we can run this through a variety of different experiments and independent assays on data sets and so on. And once the model is validated, now we can ask, okay, let's say we have a group of patients, a subset of which responded to therapy and the other subset did not. What was special about these two groups? So we can bring this information down to the level of an individual patient and then find out which network was active in that patient. So let's take a step back. So all the causal flows that we've discovered here from the cohort of patients represents all the possible dysfunctional networks across that cohort of patients. In a given patient, you wouldn't expect to see all of those dysfunctions. You would see some combinations of those dysfunctions. And so we can now leverage that information from the cohort to map the specific dysfunction in a given patient's tumor. So now we're beginning to stratify patients based on networks rather than correlates in mutations or gene expression. And so this is a much better way of stratifying the disease because you know the underlying causal mechanistic drivers of the disease. Now that information, as I mentioned, is actionable information. So you can map drugs into that network and you can then see patients who have this dysfunction are likely to respond to this therapy. And then you can design trials in a much more intelligent way where you can say, I will recruit patients who have these network dysfunctions for trials with these drugs. And so this kind of matching is called basket trials or smart trials. And they have been shown to have much better success rates. And you can recruit smaller numbers of patients and have higher rates of success. So, and then when you run these trials, you'll find out more about which patients did truly respond and who didn't. And you can then iterate and you can improve the knowledge base. Okay, so this is an adaptive learning system. So as you implement it with every patient, you learn more and you get better and better. So how does this work? Let's jump into some of the details. And if you have questions, I know you've been told to ask the questions in the chat box, but if there's a burning question, you can jump in. I'll push as far as I can go in this presentation in details, but I can jump around. So here's the idea. So there's a cohort of patients and all the patients present symptomatically in a similar way. So based on symptomatic and other broad diagnostic assays, they seem to have the same disease. But in reality, if you look at the networks, there's a subgroup of patients that share dysfunction in the same networks. So the goal is to use a data-driven way to find out who these patients are so you can then subset them and say that these patients belong to a similar subtype of disease. And through some signature analysis I will show in a minute, you can rank order them to say that, well, this patient was diagnosed early in the disease, but this patient was diagnosed later in the disease. And or you can even say this patient has a more aggressive form of the disease and this patient has a less aggressive form of the disease. So you can get some relationship regarding patients based on the signatures within their networks. How do you do that? So when you look at the multiomics profiling data sets, they're large complex matrices with a lot of missing information, noise, and there's all kinds of complexity in there. So the question is, how do you now find these modules without getting caught up with meaningless correlations? So there are ways you can do it. One is called a multi-clustering algorithm, which takes a look at all the different pieces of evidence and then connects the dots to say these 4 different independent pieces of evidence support the hypothesis that these genes are co-regulated by the same mechanisms and in the following patients. Okay? And without going into too much detail, you can see that these are gene expression patterns and these are patients being included and excluded. And you can see that the algorithm is now discovering sequence signatures, interactions, and it's essentially finding a signature of similar gene expression patterns among about 140 patients. And it is attributing that similar expression pattern to shared mechanisms that are either through transcription factor binding or microRNA regulation or through some kind of functional or physical interactions. And it tells you where the signatures are. So it can give you a lot of rich information. And this is one module, and we can discover tens of thousands of modules like that from a cohort of patients. Okay, and each of these modules can then be mined for relevance to disease. For example, as I mentioned earlier, I can take a signature gene expression pattern from a module, and I can rank order patients based on the median expression value of those genes. Okay, so as the expression trajectory increases, we see that the diagnosis of the patients, which are shown in these colors, are beginning to go from green in the lower part of this trajectory, and these are tissues from normal cadavers, and this is a brain cancer signature that I'm showing you as an example. And as you go further, you get into a more what is called a mesenchymal signature, where it is known to be a more aggressive form of the disease. So what we can do is, and just to mention, there are patients that we've excluded who we feel do not, according to the data, have the same signature. So what we can do is we can take the expression trajectory and turn it into a survival scale. And we can say, let's find out what happens to patients in the lower quintile as compared to the upper quintile with regard to survival outcome. So we can then do survival analysis like Kaplan-Meier plots and Cox proportional hazards analysis, and you'll find that in many signatures, when the patients have expression trajectory at one extreme of the trajectory, they tend to live longer than patients at the other end of the trajectory. So essentially, this expression trajectory is now stratifying patients into groups that have high and low risk. And what we've shown is the network-based approach is much more sensitive in finding such signatures. Now, once we've found from all the modules that we found, and we filtered it down to the few that have evidence of being associated with disease, we need to know what is driving the disease, what are the regulators and mutations. So we can do this in several ways. I'll give you a couple of examples. So one of the major drivers of gene expression changes in disease is through transcription factor regulation. And transcription factors are these proteins that bind in the genome to control the expression of genes. Now, the human genome is 6 billion base pairs, and these transcription factors typically recognize just about 6 to 8 nucleotides. So the likelihood of the transcription factor recognizing a sequence across the human genome is very high because there's non-specific binding. The question is, where does it actively bind? So what we can do is, what we've done is we've taken the recognition sequence of all these transcription factors in the human genome, and we've scanned it across the genome and looked for where do they have a match. Now, it doesn't mean that if there's a match, it's going to bind to that location. So we then layer in other information, independent evidence, to ask if you look across the several hundred cell types in the human genome that have been profiled for chromatin accessibility. The chromatin is folded, and when transcription factor needs to bind, it needs to open up. And there's data sets that tell you where they're open and closed. And we find that of the 3 possible binding locations, we have only 2 that are in chromatin accessible regions. Okay. All right, I'm just getting some questions. All right, we'll come back to the questions. There's nothing burning right now. So of the 3, the transcription factor can potentially bind to 2 locations. And we then can zoom in and ask, of the 2, is there evidence in any of the cell types that there was a protein actually bound? And that is called footprint analysis. So if you look at this, this is a 200 base pair resolution. You go down to 1 base pair resolution, we find that of the 2 sites that are accessible, only 1 was associated with binding of a transcription factor, putative transcription factor. So then the hypothesis would be this transcription factor is binding only to this site and not these 2 sites. And we can test this through another independent method called ChIP-seq analysis, where you can take a protein as a bait and you can, using antibodies, pull down all its binding locations in the genome and sequence to find out where it was bound. And you find out of the 3 locations we're looking at over here, it can bind only to this location, confirming this prediction. So we've done this for all the human transcription factors for which we know the recognition sequence. And we created a database that I mentioned earlier, and we put that into the knowledge base. Now, microRNA regulation works a little differently, and I'll just scream through this quickly. So microRNAs are small molecules that bind to the 3' untranslated region of transcripts. So when genes are co-regulated, they can be because of a product of both transcription factor regulation and microRNA regulation. So if you have genes in a module, you can also look to see if they share a conserved sequence in the 3' UTR. And when you find such a conserved sequence, 6 to 8 nucleotides long, you can convert that to a position-specific scoring matrix, meaning frequency of each of the 4 nucleotides in each position. And then you can create a hidden Markov model, which is essentially looking at the probability that this sequence matches to another sequence, such as sequences from a database called MIRBase, where you have known microRNA binding sequences or seed sequences. So of the thousands of microRNAs here, we can then say, this is the specific microRNA that is binding to the sequence based on the match to its PSSM. Okay. And I'll jump through this, but essentially what we've done is we have created a lot of databases that tell you which regulator might be controlling which genes. And this is called the target gene database. And when we do the multi-clustering or bi-clustering, which I showed you in the movie, what we can do is in each iteration, when you're checking to see if a gene belongs to a module or not, you can also ask if any of these binding associations are enriched in that cluster. This is called set enrichment. And when you do find that there is set enrichment, you can then use the information regarding the regulation by the discovered transcription factor or microRNA or other regulators to look for other genes that you should test in the bicluster. So the bicluster can navigate through the space of possible solutions in a guided fashion. And ultimately, when you put all of this together, you get a map like this. So I kind of given you a very quick overview of how we can take the target gene databases, combine it with transcriptome analysis and clinical assessments like survival. And we can combine this through a series of steps. And the last step is called causal inference, which I haven't shown you, but I can describe, which is the first 2 steps give you the network from regulators to modules to genes to disease phenotypes. The causal inference essentially then looks across the patient cohort to find evidence for presence of mutations that are causally perturbing this network. And there's an algorithm called network edge orienting, and we built our own algorithm to do that as well, which looks at asymmetric associations between presence of mutation and disruption of the network. So essentially you get a directional flow of information here, going from mutations down to disease phenotypes. And because we know the mechanisms here, we can also find drugs that can potentially target different points in this network. And because of the network associations, you can find drugs that might combine for a given patient. Okay. So let's take a look at glioblastoma quickly, and then I'll jump to multiple myeloma. And this is published, and so I'll give you some examples here, and if you're interested, you can go see the paper. It's in Cell Systems, published a few years ago. And in glioblastoma, the median survival, it's one of the worst cancers to get. And typically median survival is about 10 months. And the therapy typically is radiation therapy, followed by chemotherapy with the drug called temozolomide. Of course, you do resection before doing either of those things. And what you see is when you put patients through these therapies, you get about 12 to 13 month survival, so 2 to 3 month extension in survival. So it's very dismal. Now with new drugs and combination drugs, there are sporadic cases where people are living much longer. We don't understand why, but we're trying to figure it out. And so the underlying phenomenon which I described earlier is, like all other cancers, glioblastoma is also very heterogeneous. There's different kinds of gliomas, and you have glioblastoma is one of the most invasive kinds. And you have also brain metastases as well. So cancers from other tissues that can metastasize to the brain. Now we can apply the systems analysis to then figure out what are the subtypes of brain cancers and what are the networks that underlie those subtypes, and then use that to find therapies that match those subtypes of patients or cancers and patients who match to those subtypes. Now that will help quite a bit, but there's another issue which I'll touch upon, and I won't have time to go into detail, which is intratumoral heterogeneity. So even if you find therapies that are very effective on a given patient, chances are that there are some cells within that patient's tumor that might not respond to the therapy, and they could escape and then become more invasive and cause recurrence. So we need to understand the individual patient's tumor heterogeneity, all the cell types within the tumor, the heterogeneity across those cells, and then find therapies that target all the subpopulations. So when we applied SYGNAL to a patient cohort of about 422 patients, we discovered that of all the thousands of mutations that existed, about 100 mutations, 34 in genes, 68 in pathways, causally perturbed all these networks, controlled by about 100 regulators that controlled 500 modules containing 5,000 genes, representing 9 hallmarks of cancer. And when you visualize this network, it's a fairly complex graph. It's difficult to navigate in one view, but let me try to just give you a high-level overview of what you're looking at. So at the top in the pink chevrons are mutations that were discovered to be causal drivers of regulation by all these transcription factors shown as triangles and microRNA shown as diamonds. And these regulators, transcription factors and microRNAs, were mechanistically associated through that set enrichment analysis I described and a few other analyses in different combinations to each of these modules. These modules contain those genes that have similar expression trajectories and similar disease associations by function, and they connect to these different hallmarks of cancers. Now, I've annotated this to give you an insight into how we validated the model. So 6 of the mutations, 100 mutations, were previously known to be associated or driver mutations for glioblastoma. And we've identified nearly 80 to 90 additional mutations on top of what was already known. Same with transcription factors. There were quite a few that were previously known. All the shaded triangles are transcription factors that were independently associated to glioblastoma in literature or through experiments that we did to validate the model, such as doing CRISPR-Cas9 screens, where we deleted over 1,000 transcription factors and asked which ones were specifically associated with perturbing proliferation of glioblastoma cells, but not normal neuronal cells, and so on. And same with microRNAs. We did a whole bunch of experiments, targeted anti-mere microRNA overexpression, et cetera, and also in literature. So a lot of the model is statistically significant in what was previously known and recapitulated here. Same with targeted therapies. There's about 18 or so targeted therapies that have been found to work in glioblastoma, although not all of them are effective in the clinic. And targets for all of those therapies, or 18 of those therapies, were within these modules. So now we can look at this, and just to remind you, this is the graph, and we can now look at the mutations, what they affect, and so on. Let me show you one example in a little bit of detail. So there are 2 mutations here that are predicted to causally affect the control by this regulator, which is controlling a module of genes that are associated with some immune functions. So we can dig into that a little bit. And this is the network I was just showing you in the previous graph, where you have mutations in one of these 2 genes that have evidence of causally increasing the expression of this regulator, which then activates this module of genes here. And we can run a series of tests here. We can ask, do patients who have these mutations do indeed have increased expression of the regulator? And we do, when we run the test, we see evidence for that relationship. We can ask what happens with the patients who have increased expression of the regulator. Do they have increased expression of genes in this module? And you see there's a very strong correlation here. And you can then run a direct causal association analysis to ask, and this is the network edge orienting part, to know which way is the information flowing. And here what we're first doing is we're asking, do patients in mutations in these genes have increased expression of the module directly from here to here? And we see a strong association. And finally, to confirm the information is going through IRF1, we remove patients that have increased IRF1 expression and then ask, do we still see this association? The answer is no. So that tells you that the mutation information or causal effect is flowing in this direction. Okay. Now we can look into the functions of genes in the module, and we see that those genes are involved in functions of antigen processing and presentation, and they're shown in all these red font proteins here in this schematic. And the hypothesis based on this analysis was patients who have this perturbed network will have altered antigen presentation, so their tumor microenvironment should be significantly perturbed. So we looked at the tumor lymphocyte infiltration into, or T cell infiltration into the tumor, and we found that in patients where the network was perturbed, there was moderate to severe infiltration by T lymphocytes. So this essentially validates that the tumor microenvironment is altered in patients that have these mutations, and we have a hypothesis about how that is happening. So we can target that network. And as an example, in a different study, we worked with Mike Jensen, whose company Juno Therapeutics was bought out by Celgene for $9 billion. He told us that the only immunotherapy trial that was run in glioblastoma was against this receptor, IL-13 receptor alpha 2. And the patient, there was a single patient who responded really well, but when the patient had a recurrence, it was because the expression of the receptor had gone down. And the question he asked us was, can you use a network analysis to find a drug that could induce the expression of the receptor? So we did the analysis, found this network, and found this drug that could putatively block this flow of information to then induce expression. And then we showed across the tumor cell population in that study that we can now use cholesterol to, in a precise way, increase the expression of the receptor. Just as an example of how we can now use a precision approach, and you can even tailor it to patients who have mutations in interferon gamma pathway, for example. So the way it could work for glioblastoma is we could now map patients into each of these modules. For example, if a patient maps to a particular module in the upper quintile, we can then decipher what might be the clinical outcome for that patient based on how the group of patients performed. And we can say that the patient is at higher risk because these patients did not live as long. And we can also jump into the network around the module and say exactly what are the genes in the module and what is controlling the expression of the gene trajectory. And we can find drugs or microRNA inhibitors, and we can do investigational and drug discovery type analysis, or we can take FDA approved therapies and match them to the network. And then we can run these assays, and this is real data where we've taken a microRNA mimic to perturb a microRNA controlling the network of this module. And we've taken a drug that also targets the same network and identified the IC50 for the two agents, and then done a checkerboard matrix to show that romidepsin, which is effective in stopping proliferation of cancer cells but toxic, can be effective at a lower dose when we combine it with microRNA mimic 4863P. And this is likely to work only in patients who have this perturbed network. Okay. So as I mentioned, this work is published. I'll come back to how this goes to the clinic, hopefully towards the end. And now very quickly in terms of the intratumoral heterogeneity. So this is for a single patient. So we're working with the Swedish Neuroscience Institute, where when a patient comes in and is diagnosed with brain cancer, they do a biopsy, and from the biopsy, they then harvest what are called glioma stem-like cells. And the theory there is when there is recurrence, it's these cells that are responsible for driving the recurrence. And if we know something about these cells, we can then tailor a cocktail therapy. So when the patient has a recurrence, we can give the therapy to the patient. And it has been shown by our collaborators that when you take this approach in 5 patients out of 30 that they have enrolled into the trial, who received a triple drug regimen, their survival has gone up to about 48 months. So from 12-month median survival to 48 months. So dramatic improvement. But we still need to figure out what is the best way to combine the therapies, because the space of combinations is very large. So here what we're looking at is the patient-derived glioma stem-like cells, or PDGSCs, over a time course experiment. 0 is at the early stage of the experiment. And then these are over 3 days. And for one drug in a panel of 76 drugs, what we find is when you use this drug called pitavastatin, which is a cholesterol pathway inhibitor, you see the cells are changing in state. This is a two-dimensional projection in the UMAP space. And we can then annotate the cells based on subtype. And you see that there's classical to proneural to mesenchymal. And mesenchymal is the most aggressive subtype. And we can then apply something called a trajectory analysis. And there's several ways you can do it, including monocle 3, RNA velocity developed by other groups. We haven't developed this. And we can find pseudotime trajectories. So this is essentially building a trajectory through this dynamic response to say, predict what might have been the directions in which the gene expression trajectories changed to drive the cells from a classical state to a much more mesenchymal state. And so this is the principal path. So you see it's going from proneural to proneural/classical subtype all the way to mesenchymal. And what we can do is we can align the cells along this trajectory. We can apply the signal network analysis, and we can then decipher what are the regulatory networks that are changing along the trajectory, and what are the regulons or regulators that are driving this shift in the cancer cell phenotype. And we can identify very specific players that are driving this and create a flow, a causal mechanistic flow map. In this case, we see that we predict that these are the regulators that are acting in sequence to drive the cells to escape from therapy and become more aggressive. And along the trajectory is a player that we can target with a drug called genistein, which is used in other cancers. And so now our hypothesis would be combine pitavastatin with genistein. In fact, do that over some temporal regimen where you give the patients pitavastatin, and then within a day or so, you then give them genistein, or do that together. And these are things that we are now looking into at a fairly early stage. So let's, for over 10 minutes, just quickly go into multiple myeloma, just to show that what I've shown you is not restricted to just glioblastoma. And the glioblastoma work was funded by NIH and through partnerships with different collaborators. And the multiple myeloma study has been supported by collaboration with Celgene, which was brought out by Bristol Myers Squibb, and also through a Washington Research Foundation, who has given us money to translate this and the glioblastoma model into the clinic. And these are all our collaborators. So same idea here. I won't go into how we build the network, but we've advanced the methodology when we worked on multiple myeloma because it's a more recent study we've done. And here I want to introduce you to some new concepts. So I've been referring to this box here as a module, and here onwards I'll call it a regulon. So regulon is a set of genes that are co-regulated. And program is a set of regulons that have similar activity profiles. It'll become clear what I mean in the next slide. And patients who have similar programs belong to the same state. So when we run this on about 880 patients with multiple myeloma, and this data set came from the Multiple Myeloma Research Foundation, we find that these patients have a disease that is driven by 124 causal mutations. So there's thousands of mutations across the patients, and these are the summary of 700 odd somatic mutations that are found recurring across the patients, but only 124 are causal drivers. And we find out exactly how they're acting through which regulators of what regulons, how many genes belong to the regulons. And here we can do something which we haven't described in the previous glioblastoma study, is we can now look at the flow of information, every single flow from a mutation down to a gene, to a regulon, and find out there's 13,000 possible flows. And which is very complex, but interestingly, you can now cluster the causal flows, and you find, or the regulons, you find transcriptional programs. There's just 140 transcriptional programs that describe multiple myeloma. And of the 141 transcriptional programs, you can find that they can be clustered into 25 distinct states, of which 5 are very high risk. So we've taken this very complex set of information and converted that into a structured map that can predict clinical phenotype. So here are all the patients that I just described. These are all the 3,000 odd regulons. And you can see that regulons that are behaving similarly across patients are grouped together into program. And then patients who have similar program activity profiles are in the same state. And what we can show is if you take a program and you look at its on-off behavior across patients, that's actually a good predictor of survival. So patients in which a program is active may do poorly than patients in which that same program is inactive. Okay, and we've done that for every single program. We've done it down to every single regulon. The states are even more predictive. So patients belonging to the same state have very similar clinical outcomes. Okay, and here's a summary of all the states, the 25 roughly states that we've identified, ordered by risk. And each dot here is a patient. Okay, and I'll give you one example. So in multiple myeloma, cytogenetics is used. So translocation of certain chromosomal segments are predictive of risk. For example, T414 is believed to be very high risk, whereas T1114 is believed to be relatively low risk. But if you look at the distribution of patients across these states, you find that for T414, which is supposed to be very high risk, you have patients who are also low risk. And the other way around as well, T1114, which is believed to be moderate to low risk, you have some patients of very high risk. So the stratification based on cytogenetic markers alone is not accurate. And I'll skip this here. Just want to give you one example. For example, T414 patients, we can look into their program activities and we find that among T414 patients, when program 68 is inactive, it's low risk. You have median survival, progression-free survival, or PFS of 30 months. But if program 68 is active, then the median survival drops to about 22 months. But within that group, you have one that has the 22-month survival outcome. But there's another where the program 61, which is unchanged or active among the lower risk group here. If the program becomes inactive, then the median survival drops to 5 months. So these patients with T414, program 68 on, program 68 off, are very, very high risk. And you can find the underlying functions. And so we know how to identify this subgroup of patients. And here's the survival outcome of these patients. It's quite striking. And so each of these, so here's the T414 group that I described. And we can now within each group find multiple subtypes. And those are shown here in these rows, ordered by risk. Okay, so for all the cytogenetic markers, we now created a much higher resolution subtyping map. And for each subtype, we can then look at the activity of programs that contain targets of known drugs. These are all the targets of known or currently used multiple myeloma therapies. These are therapies that are currently being tried in clinical trials, and these are completely new that we're predicting should be tried. Okay, and we're telling you which patients they're likely to work on. And so it's not just taking the therapy and trying it on all multiple myeloma patients, but use it on a very specific subtype of patients who have certain markers. And here's an example. So patients who relapse typically have escape therapies that target these targets. Okay, and at baseline and relapse, we can look at what is the expression of these targets across these patients. And you see each of these targets has no difference in expression when patients relapse. But if you look at the network activity, you see that most of these networks have become downregulated, okay, upon escape. Whereas the target for this drug, which is used at relapse, typically carfilzomib, has been upregulated. So essentially, we can explain why patients are escaping therapy. And then we can look at all the programs that are being turned on and off. And the most turned off program across all patients is the program that is targeted by immune modulators or IMiDs, which is used across all multiple myeloma patients. So these patients have completely turned off this program so they can escape that therapy. And instead, they're turned on some other programs. And we can look at what these programs are. And then based on the genes in those programs, we can now pull out the network associated with those programs and find out what drugs are likely to work. And so we found about 12 druggable targets in the network gets turned on in multiple myeloma. At relapse, identified about 17 FDA-approved therapies that can target each of these programs, and found that 10 out of the 12 are in the network called the B cell maturation antigen network, or BCMA network. And there's an investigational therapy called GSK, and that long number, by GlaxoSmithKline, which they found through brute force. But again, they don't know which combinations of these druggable targets are active in which patients. So we can now tailor targeted therapies plus the BCMA drug based on the patient network signatures. So for a given patient, as I said, you don't have all of the network active, and we need to find out which combinations of mutations are active, whether they are acting through the causal flows that we've discovered, and so on. And here's an example for an N of 1 analysis we did, where we could look at all the possible mutations in the patient, and the ones shown in red are the causal flows that were found to be active in that given patient. So we found these mutations, and we found evidence that these regulons are putatively active. And accordingly, we can pick any of these drugs. And of course, there's a lot of other types of information that could be used. So again, this paper is published, and well, it's in bioRxiv right now. It's in review in a Nature journal right now. Hopefully, we'll have it out in print soon, but you can still access the data. So in the last slide, I just want to give you a vision of how we are now applying this in the clinic. So we're working with the Fred Hutch Cancer Center, UW Medicine, Swedish Neuroscience Institute, and soon with others as well, to do the following. Is when a patient comes in into the clinic, we can, typically what happens is you get a biopsy, the biopsy is biobanked, you do molecular profiling. In cases like multiple myeloma, you may get cytogenetic markers, and we can inject the platform we've developed right in the middle here. So you can then take all these profiles from the patient and then analyze it with the signal network that we developed, score the networks, and identify specifically for this patient what is the network that is active, or what sets of networks are active. And then through a combination of analysis across drug databases, we can then come up with a list of therapies that are likely to work on this patient with all the evidence for why we think each therapy or therapy cocktail is likely to work. Now, this will be taking thousands, tens of thousands of papers and data sets, etc., and condensing that, crystallizing that into one report, because clinicians have only 10 to 15 minutes typically to review a patient's report. So they can't go and read all the literature and all the complex information. So this is meant to help bring everything to their fingertips through interactive interfaces. And this can go to a tumor board, which is typically a group of people, different numbers of people sitting together, and they have different expertise. They look at the evidence, and they can then decide exactly what drug should be given to that patient. So this whole thing will require many different pieces to be connected, not just the science that I described, but also all the complexity of dealing with electronic medical records, getting profiling done in a timely manner, the ability to then analyze safety information, clinical trial information, and then rank prioritizing it for a given patient. Okay. So I just leave the slide here and just wanted to thank a whole bunch of people. I might have missed a few, but funding from a variety of different sources. For glioblastoma, there's a whole group of people. Chris was the person who wrote the first paper, but many of these other people have contributed to the experiments. James is doing the single cell analysis. Kavya is doing the clinical translation analysis with UW. With Anu Patel, we are looking at tracking individual patients from primary biopsy through avatars in mice and tumor slice cultures down to autopsy, how tumors respond to therapy. So we can become much more targeted in doing a therapy plan for individual patients. For multiple myeloma, Matt Wall was the main person driving the whole effort. There's several others. All for Cure is the group that is built this whole system to track individual patients and create a virtual tumor board. And the validation for multiple myeloma we're doing in collaboration with David Coffey and Pam Becker, who are physicians at the UW Hutch Cancer Center. And I should mention Charles Kops and Parvi Hoti are the Swedish Neuroscience Research Institute, and they're working on the glioblastoma clinical validation. And we're translating this actively now with help from Sardar and Ashutosh. And just wanted to mention that we've worked for some time with PSL in persistent systems in applying some of these ideas to breast cancer. And we've also been discussing with them ways that we can partner with them to go and execute on these ideas in large hospital systems. So I'll stop and I'll be happy to take questions at this point.
Dr. Baliga, thank you so much. This is more than enlightening. This is, to know that such specific work and such comprehensive work is happening, at your lab and everywhere across, is really enlightening. There are many questions and we have very less time. So what I've tried to do is to try to take more questions and combine a couple of them. My first question would have been how it works in a clinical setting, because a lot of people have identified their own conditions and they've spoken about what they have gone through during cancer, and probably what they were trying to ask is, how does it work in a clinical setting, which probably you gave in the last slide, which was a very excellent snapshot. But what I would want a little bit clarity for the audience here is that, which are those specific cancer types wherein precision medicine has actually made a change, so that people know that apart from the routine therapies that they are taking and they have been provided at different centers, where is more hope for them? So if we can.
Yeah, so I think there are a few cancers where precision medicine has worked really well. I gave one example in Herceptin for breast cancer early on. So patients who have the HER2 mutations tend to do really well if they receive Herceptin. And but that's a small group of patients with breast cancer who tend to have that kind of mutations. Now, and there's other criteria also that physicians look at in nodal involvement and quite a few things, which is outside my area of expertise to go into details there. But also in case of melanoma with the BRAF inhibitors, when patients have the V600E mutation in BRAF, they tend to respond well to verapamib. And now, even the precision therapies tend to work really well, but you still have the risk of recurrence and relapse. And so I think there's more and more examples coming up, but a lot of the precision therapy, in fact, most or all, have to do with finding mutations that are predictive, and there are panels. In a clinical setting, what happens is the patient gets biopsied, and then the biopsy is sent out for profiling. And many places are doing this, but not all. So I think patients need to advocate for themselves to say, I would like to get profiling done. Of course, you have to figure out how to pay for it and things like that. And
Yes.
That can get expensive. And there's different profiling companies that offer these services. In the US, you have quite a few of them, including Foundation Medicine, there's Caris, there's also Tempus and Guardant Health. And so all of these companies have different platforms, and they specialize in some or all cancers. Caris, for example, who I know a little better, have a really state-of-the-art facility in Arizona where they will take the patient's tumor biopsy. It comes to them via FedEx, and then they have a whole process to kind of QC the sample, make sure that there is not too much necrotic tissue in the tumor slices that they do from the PFFE embedded biopsy. And then they profile it with sequencing, and they have a report that looks at about 1,000 mutations and gives you some predictions. And they have about 70 odd drugs that they can then map based on the mutations. And they just send that to the clinician. So the challenge in the clinic is actually getting the profiling done in a timely manner.
Right. So you touched upon melanoma and breast cancer, right? And a couple of questions which also came more from lung cancer, but people have not really specified which kind of lung cancer, but lung cancer, brain cancer, as well as leukemias. So if you have some insights on those three also, then I'll probably be able to answer those three individuals to you.
I would rather not go into those specifics. I mean, I haven't worked on those cancers directly myself. I mean, I don't want to give misleading information. I mean, for brain cancer and for myeloma, we have done most of our work. But I can certainly look into those. If you can forward those questions to me, I can certainly forward it to experts that I know who might be able to answer those
That would be
questions.
fantastic. So I'm quickly going to go forward because I have a long list. Also, there questions that I receive are on survival of patients. So how does precision medicine, the median survival rate improves with precision medicine as compared to usual therapy? How much would that in certain cases?
Right. So the example I gave and my first-hand experience is through my collaborators at Swedish. So what they do is, as I mentioned, they take the patient-derived glioma stem-like cells and they run a high-throughput screen with 76 drugs. And they look to see which of those drugs are potentially effective for against a given patient's tumor cells. And from the single agent screen, and that is done each drug, 8 replicates, multiple doses, and it's a fairly elaborate screen that they run. And then they find from that panel a list of drugs that are effective, and they combine them in a 3-drug regimen. So this step is not currently not model driven, because even if 2 drugs may be effective independently, together they may work against each other. So they have to run what is called a combination screen analysis to look for a synergy or antagony and things like that. Now that space of combination is exceptionally large. So if 10 drugs have worked, for the 76 drugs, you have to run about 1.5 million tests, and there's not enough tumor tissue to run that. So there are some challenges there. Even then, in the trial, they so far looked at 30-odd patients. 5 of the patients have gone through the whole process and they come back with recurrence, and they've received triple drug regimen. So they picked 3 of the drugs that did work, and those patients, their median survival have what would have been expected to be about 12.6 or 12.8 months is variable, but has been positive for all of them, with some living as long as 48 months. So it's a 4-fold increase in survival.
Yes, that is drastic, actually.
Yeah.
I'm going to take the last 2 questions. One is, what is the use of AI in detection of the type of cancer as well as the therapy, which I'm guessing people want to ask is the precision therapy in that sense. Probably AI and the ML bit in this.
Yeah, AI, ML, I mean, these are real buzzwords. I mean, they mean so many different things, and they can, look, people have tried, and there are certain things that they do really well. I mean, in terms of diagnosis, if you look at things like radiography, and the human eye can make errors, and with AI and machine learning, you can do much better job in classifying those images, perhaps. Actually, they can. I mean, you can also use AI to detect tuberculosis in cough, for example, before even it's symptomatic. So there's a lot of good applications. And when it gets down to selecting therapies, what AI and ML work on is having information about which therapies are likely to work from a cohort that has already received the therapies. What you're trying to find with AI and ML is what are the signatures that are stratifying the patients who did respond from those who did not. And so for applications like that, if you really don't care why the patients responded or not, you could apply that. I mean, there's value in doing that. And typically these are black box approaches, meaning when they do find a classifier, oftentimes you can't explain why these patients responded. You can just predict that they will. And where it becomes challenging is when you want to repurpose therapies. So to repurpose therapies, you need to really look at you don't have cohorts of patients who receive the therapy, because the therapy is used in some other indication. So when you don't have the data set to begin with, you can't do AI and machine learning. So for that, you need causal mechanistic understanding of the disease. So you need a theory-based approach more than a brute force data science-based approach. And what we're trying to do is we're trying to marry the virtues of both approaches in a way where we can find prior knowledge to guide the AI and ML and statistical inference approaches, so we can find new information also, not just be limited by what is known in literature.
My last question is, and I would really want to apologize to everyone, we may not be able to take up questions here, but we can try and get back to you. And some of the questions are repetitions, so it may not specifically answer your question, but gives you information around your question. So my last question to you, Dr. Baliga, is that there's a lot of questions on gene panels. So there are students, they would want to know how to approach developing a gene panel, and what would be the role in prognostic and as well as diagnostic markers in that situation.
Yeah, so there's multi-biomarker panels that can look at mutations, expression, microRNAs, and so on. Now, the notion of panel has to be understood. I mean, panel could mean just a slide on which you have some probes that you can use. Panel can also be a virtual panel, a digital panel, where you run a series of different technologies. Cytogenetics would look at a different way of looking at chromosomal translocations. You can look at sequencing with RNA sequencing or microarray analysis. RNA sequencing is now the predominant way it's being done. You have bisulfite sequencing for methylation. So there are different technologies that can profile in a genome-wide manner the changes that could be happening in tumor cells. Now, panel would refer to the subset of data within those large amounts of data sets generated that have predictive value. So you can say there's a 13 gene biomarker panel. So that 13 genes, it could be expression levels of some, mutations in some, and methylation of others. There's enormous value in doing that in terms of, but remember, these are diagnostics in the end. So you need to find genes that not only can stratify. So telling someone you are high risk is great because it'll help them make a lot of decisions. But if you can follow that up by saying, because X and Y is dysfunctional, that is somewhat helpful. But if you can say, and we have a therapy that is likely to work, and here's a trial that is being run over. So there's several steps that you really need to think about that layer on top of just building a panel. And that brings in a level of complexity. But coming from the side of clinical outcome is always a good idea to say, if you want to improve clinical outcome, panels become a part of that solution, but there's many other steps that you will have to consider to make sure the whole solution is then beneficial to a given patient.
That is really helpful because, unless, I mean, as you're saying that, knowing something where you really can't do anything may not be as helpful. You want to focus on areas where there is actually something that you can do when you're creating a panel like that. So Dr. Baliga, this was enlightening. I know that the time at which that you've come to give us this talk is a very odd early morning, and most of us would be very peacefully sleeping like babies at that time. But thank you so much for taking the effort. And I would, from the entire, more than 300 people who logged on to listen to you, and the entire Manu team, would sincerely thank you for being a part of this webinar series. So thank you everyone. Have a good day and a good evening. We can all go back to sleep. And Dr. Baliga, I think you have a whole day to work ahead. So you have a good day. All right.
You're welcome, and it's been a pleasure, and I enjoy doing this. So I'm glad you reached out, and I look forward to interacting with you and others as well.
Thank you so much. All right then, everyone.